everyone, welcome to Chapter 3, Biological Macromolecules. So this is going to tie in a lot to the chemistry chapter. This will tie in a lot to our chemistry chapter that we did this week as well. We'll be talking a lot about our macromolecules. I'm sure that you've heard of this before. And maybe if you're really into nutrition or you're very active, you tend to keep track of your macromolecules in your diet. So we'll talk a lot about the nutritional aspect of some of these macromolecules, as well as how they may affect plants and animals alike. So our macromolecules are polymers. They're large molecules. Polymers are made up of monomers. So monomers are like building blocks. They're typically smaller organic molecules that are built together to form polymers. For each specific polymer, there's generally a specifically named bond for that polymer, for that macromolecule. So as we go through and talk about each of these macromolecules, pay attention to the named bonds for each one. Our four major macromolecules are proteins, carbohydrates, nucleic acids, and lipids. Those are our polymers. Amino acids make up proteins, so amino acids are the monomer for proteins. Monosaccharides are the monomers for carbohydrates. Nucleotides are the monomers for nucleic acids, and fatty acids are the monomers for lipids. Those will be extremely important. First, let's talk about how some of these molecules might be coming together. So there are two different types of bonds that are very similar, that are often implicated in the formation of these macromolecules. So they can be formed via dehydration synthesis, which is when monomers are going to combine to form covalent bonds. They come together to form these polymers, and as a byproduct, they release water. So remember, if you're dehydrated, you don't have enough water, so you release water to make a larger molecule in this case. On the other hand, hydrolysis is taking a bigger molecule and breaking it down using water. So hydro means water, lysis means to break. So to break with water, we're taking a larger molecule and using water to break it into smaller components. Both dehydration synthesis and hydrolysis reactions can be catalyzed by specific enzymes. We'll talk a lot about that when we get to proteins, because enzymes are a type of protein. And to catalyze a reaction such as dehydration synthesis or hydrolysis reactions, that means to speed it up. And that's the main job of enzymes, to catalyze reactions. So carbohydrates are the first macromolecule that we're going to talk about. So remember, we said that the monomer for carbohydrates is monosaccharides, simple sugars. And we'll talk more about monosaccharides and how they combine to form disaccharides and also polysaccharides. Mono meaning one, di meaning two, and poly meaning many. Saccharide is a generic for sugar, so one sugar, two sugars, many sugars. Many people don't know that carbohydrates are essential for a healthy diet for humans, and most animals in general. Carbohydrates should make up 45 to 65% of your diet, which is a larger amount than most people realize, because oftentimes carbohydrates are given a bad rap of being unhealthy and contributing to weight gain. But there are very healthy forms of carbohydrates in the forms of plants. They don't have to be simple sugars and grains that are unhealthy for you. They can come in the form of fiber as well. So just keep in mind that carbohydrates aren't a bad thing. Listed here is the stoichiometric formula for carbohydrates. So this just shows the ratio of carbons to hydrogens to oxygens. So N stands for the number of carbons in a carbohydrate. Each carbohydrate will be different in that sense, but just know that its ratio is 1 carbon to 2 hydrogens to 1 oxygen, and that will be standard for all carbohydrates. So let's talk about monosaccharides, the building blocks of carbohydrates. They're typically known as simple sugars. Some examples are glucose, galactose, and fructose. Typically in length, they are 3 to 7 carbons long. They can either have an aldehyde functional group and become an aldose, or have a ketone functioning group and become a ketose. Generally, these monosaccharides are going to be hexoses, so they'll have this C6H12O6 formula. Because they all have the same formula, that makes them isomers. Monosaccharides are really important as energy sources in plants and animals. So looking at these glucose, galactose, and fructose, I'll give you a second, but try to determine which ones are ketoses and which ones are aldoses. So aldoses have that aldehyde functional group, which is that double bond to an oxygen and a hydrogen at the end of a molecule. If it's sitting at the end, such as in galactose and glucose, that makes them aldoses. Ketones have a double bond to an oxygen somewhere in the middle of the molecule, not on either end, so that would make fructose a ketose. Monosaccharides join together to form disaccharides. To become a disaccharide, they have to undergo dehydration synthesis, and then the bond that is specific to disaccharides or to monosaccharides to make larger molecules is a glycosidic bond. It can either be an alpha or beta one, and that just determines how the carbon is positioned. The position of the bond and which carbons it attaches to between the two sugars. Some examples of disaccharides are sucrose, which is the combination of glucose and fructose, lactose, which is the combination of glucose and galactose, and maltose, which is 2 glucose molecules bonded together. So over here on the side you see a glucose molecule coming together with the fructose to make a sucrose. And here's the glycosidic bond right in the middle. So if you are somebody who is lactose intolerant, we have digestive enzymes to break down these sugars, and they're all specific to each sugar. We'll learn later that enzymes typically have the suffix of ase. So lactase breaks down lactose, and if you are lactose intolerant, that means that either you don't have enough lactase enzymes or they are not as receptive to lactose molecules as they should be. Polysaccharides are going to be these long chains of monosaccharides that are still linked together by those glycosidic bonds. Some examples of polysaccharides are glycogen. Glycogen is the storage form of glucose that's found in the body in humans and animals. So if your blood sugar is ever low, oftentimes your body will break down that glycogen and it stores in the muscle and the liver to use blood sugar throughout your body in times where you're not eating. In plants are going to store starch as amylose or amylopectin. This is going to provide some food for seed embryos. When humans are eating plants that are very starchy, we have enzymes that help to break down that starch into glucose, which is that usable form of sugar for us. And those are amylase enzymes. So amylose is broken down by amylase. Cellulose is also a polysaccharide. It's found in plant cell walls. It's in really high concentration in wood and paper. That's what gives it its tougher texture. Cellulose also makes cell walls in plant cells rigid for the protection of the cell in the plant. Humans can't break down cellulose, but herbivores can. So when we eat plants as humans, that cellulose is used more as an insoluble fiber than it is a nutrient, but in herbivores it's actually used as a nutrient because they have an enzyme called cellulase that helps to break down that cellulose. Termites are pretty cool too because they have these protozoans that live in their digestive system that help to break down wood, which, as we just spoke about, is really high in cellulose. So without these protozoans in their digestive tract, termites would die because they wouldn't be able to break down that wood for a usable source of energy. Chitin, I know it looks like it's not pronounced that way, but it's chitin. It's found in the exoskeletons of arthropods like insects and crustaceans. It's also found in the cell wall of fungi. It also acts as an insoluble fiber for humans and animals, sort of like cellulose. Insoluble fiber is great for us because it helps to lower blood pressure and cholesterol. It's really heart healthy. So it's found in crab and lobster, also shrimp, but unless you're getting a locally farmed shrimp, I wouldn't recommend consuming shrimp all that often because it's really not sustainable for the environment. So starch, glycogen, cellulose, chitin, they all have alpha 1 to 4 or alpha 1 to 6 glycosidic bonds. So remember I said the alpha and the beta is just going to tell you the position of where these carbons are. Chemists like to name these carbons or number these carbons 1 through 6. So it just tells you that the bond is between carbon 1 on one sugar and carbon 4 on another, or carbon 1 and carbon 6. So we already talked a little bit about not being able to break down those linkages in cellulose and chitin. So carbohydrates have always been a part of diets of humans and humanoid type creatures, as far back as our earliest ancestors are concerned. It is really important for our energy sources, and we'll talk a little bit about that when we get to the cell chapter and how we kind of harness that glucose for energy. This has some implications in the ketogenic diets. They do work pretty well for some individuals because they use more amino acids and ketones for energy rather than sugars. So we'll talk a little bit about that next week and their implications when we get into those different pathways. So like I said, fiber, either soluble or insoluble, are really important in diets. It helps to promote bowel health, prevent colon cancer, lower cholesterol levels, very heart healthy, and it also lowers your blood pressure. Okay, so that was carbohydrates. Let's shift into lipids. So we talked about how lipids are nonpolar and hydrophobic this week. They don't mix well with water. They provide a good source of insulation, particularly in aquatic birds and mammals. That helps to keep them dry. In aquatic birds, it helps give them a protective layer of lipids on their feathers. so that water droplets kind of just cascade off. Lipids are also used for a storage of long-term energy. They're a building block for a lot of different hormones, especially those steroid hormones that we'll talk about in just a few minutes. So I'll talk about each type: the fats, the oils, waxes, phospholipids, and steroids. So first up is the fatty acids, triglycerides. They're also known as triacylglycerols. Same thing, different wording. It's the combination of glycerol and fatty acids. Fatty acids can either be saturated or unsaturated. When they are saturated, that means every single carbon is attached to a hydrogen, and that there are only single bonds. The one on the aldehyde group does not count. They're talking about strictly within this hydrocarbon structure. Now, when it comes to unsaturated fatty acids, that means that there is at least one double bond between carbons present. If there's one double bond present, that's called a monounsaturated fatty acid. If there's more than one, then it is a polyunsaturated fatty acid. Our unsaturated fatty acids are better for us, and we'll talk about that in just a second. But you can see here that there is a double bond between these carbons, so it's not completely saturated with hydrogens like it is in this previous photo. Saturated fatty acids tend to be solid at room temperature. A good example is butter. Butter is a saturated fatty acid. Unsaturated fatty acids tend to be liquid at room temperatures. Think of your cooking oils. Those tend to be mono or polyunsaturated fatty acids. Coconut oil is another example of saturated. And we also have trans fats, which we also talked a little bit about this week. So they have a different type of structure. You can see that these hydrogen groups on either side of the double bond are flipped. So that's what makes it a trans fatty acid, or a trans fat. So you can see that both in saturated and in the trans fats, they don't have double bonds that make them have this kink in the structure. This is important because as they accumulate within our bodies, less can accumulate if they form this kink in their structure than if they are linear, which has large implications in accumulations in our arteries and excessive fat deposits. Alright, so let's talk about omega fatty acids. There are some essential fatty acids that you need, and they have to be obtained from the diet. These are our omega-3s and our omega-6s. Omega-3 fatty acids, some examples are alpha-linoleic acid, ALA, EPA, and DHA. Good sources of those: salmon, trout, tuna. If you don't like seafood, you can take fish oil supplements, and it'll do the same trick. Won't give you quite as much protein as doing the fish will, but it will still get you those essential fatty acids. Omega-3s help to lower your chances of heart attack. They reduce your cholesterol, blood pressure, prevent blood clots, lower inflammation, and reduce your risk of cancer. All amazing things. There are a couple of signs of deficiency for omega-3s. Having dry skin, poor concentration, or just a couple. Those who take fish oil supplements or eat a lot of a fish-based diet tend to feel like they have a lot more energy. Omega-6 fatty acids help to stimulate skin and hair growth, maintain bone health, regulate metabolism, and maintain your reproductive system. Sources for these are nuts and seeds, soybeans, and eggs, and there's a ton more, but those are our primary sources. Some signs of deficiency of omega-6 are bumps on the back of your upper arms. Those are called follicular keratosis, sometimes keratosis pilaris. If you have dull, kind of lifeless looking hair or skin, dandruff, or brittle nails that tend to break easily. So, best case scenario, you take these essential fatty acids in through diet, but you can also supplement these. There are waxes. They help to coat bird feathers and plant surfaces as an extra layer of protection and waterproofing. Phospholipids are the major components of our plasma membranes. So they are extremely important. Plasma membranes are the outer surfaces of our cells. They are known as amphipathic because they have both hydrophobic and hydrophilic parts. In the cell, they have a phospholipid bilayer. The inner parts of the bilayer are the hydrophobic tails of the phospholipids, and the outer portions are these hydrophilic heads. So this helps to bring in polar molecules and keep out nonpolar molecules unless there is a channel in which they can pass. The most important part is keeping liquids out of the cell that shouldn't be in the cell and keeping all the liquids in the cell that should be in the cell. Otherwise, the cell would combust. When phospholipids are by themselves in water, they spontaneously form micelles. Micelles have this hydrophilic outside and hydrophobic inside, but instead of a linear structure that's shown here, it forms a sphere. So micelles are found a lot nowadays in face washes, also in soaps. And they're really great at being emulsifiers and getting rid of fat and lipids. So when you're cleaning a pan, you can't just use water to clean it, right? You have to use soap in order to get those oils and greases and fats off of that pan. Same goes for face washes. If you've got a lot of oil on your face from the day, it helps to really get that off of your skin. So these micelles tend to surround the nonpolar substances, act as emulsifiers to help break that down. Steroids are also a type of lipid. They have these fused ring structures. They're hydrophobic and insoluble, just like all of the other lipids. They tend to have these hydroxyl functional groups to make them alcohols or sterols. And the most common of the steroids is cholesterol. Cholesterol gets a pretty bad rap because you hear a lot of times that things are high in cholesterol, or you're trying to avoid cholesterol or lower your cholesterol, but there are certain types of cholesterol that are really beneficial for you, and your HDLs, your high-density lipoproteins, are your good cholesterol. They help to prevent you from having heart disease. So they are extremely important. It's synthesized by the liver. It's also a precursor to estrogen and testosterone, so without a lot of cholesterol, it would be difficult to make those hormones. It's also a precursor to vitamin D and bile salts. So bile salts are made in the liver, stored in the gallbladder to help break down fats in your body that you take in from your diet. And vitamin D is obviously an extremely important vitamin for strong bones, teeth, and muscles. Proteins are our most abundant macromolecule, and it's also the most diverse of all the macromolecules. They can function as enzymes, hormones, toxins, transporters, even storage. There's so many more different functions for proteins. The shape of the protein is critical to its function. Proteins are very diverse in their shapes, and not all proteins look alike. Most proteins have an ideal temperature or pH that they like to be in, survive in. So any changes to temperature, pH, or exposure to chemicals can lead to denaturation or the protein turning off and not being able to function. One type of protein is enzymes. Enzymes are extremely important. Their job is to catalyze biochemical reactions to speed them up, lower the activation energy in order for them to be able to proceed from reactant to product quickly. Enzymes are specific to the substrate. Generally, they are named after the thing that they are breaking down or speeding up. There are catabolic enzymes. Catabolic, think like catastrophe, or enzymes are taking a large molecule, and breaking it down into smaller pieces. The sum of all of the chemical reactions in the body that are breaking down larger molecules into smaller constituents is known as catabolism, which is a main part of metabolism. Anabolic enzymes are taking smaller constituents and making them into larger molecules. So think of things like anabolic steroids. We're helping to create something larger, like larger muscle mass. So all of the chemical reactions in the body that are coming together to form larger molecules is known as anabolism, which is the second half of metabolism. So anabolism and catabolism come together to form metabolism, which generally stated is just the sum of all of the chemical reactions that are occurring within the body. And then most enzymes are catalytic enzymes. They help to speed up the reactions. Hormones are chemical signaling molecules. They generally target a specific tissue or organ in the body. They tend to have long-lasting effects in the area in which they're targeting. Hormones can be protein-based. They can also be steroid or lipid-based. They tend to control or regulate physiological processes such as growth, reproduction, and metabolism. One really good example of a protein-based hormone is insulin to help lower our blood sugar. Here's some more examples of protein types and functions. And remember that the monomers of proteins are amino acids. Amino acids are named such because they have an amino group and a carboxyl group, and when you have a carboxyl group, that makes you an acid. So, amino acid. There are 20 common amino acids found in proteins, 9 of which are essential, so you don't make them in your body. You have to get them from your diet. The R group on the amino acid will determine its chemical nature and properties. Their specific type of bond is called the peptide bond. When many amino acids join together via peptide bonds, they form polypeptides, and many polypeptides together form proteins. Once a protein, they have a unique name and a unique function. The structure of the protein is critical to the function. There are primary, secondary, tertiary, and quaternary structures of proteins. They don't always have all 4. Sometimes they only have 1, sometimes they only have 2, 3, 4, etc. The primary structure of the protein is just the unique sequence of amino acids. How they fall together in a line determines their function. This is determined by genetic encoding from genes, from DNA. Any change in sequence due to any genetic mutations is implicated in a lot of diseases. One really good example of that is sickle cell anemia. When a mutation occurs in the primary structure of amino acids for that protein, the red blood cells form a sickle shape rather than their typical biconcave structure. This allows them to get lodged in blood vessels rather than passing freely through in a singular line. This can cause a lot of health crises, blood clotting, and so many more complications. The secondary structure of proteins involve alpha helices, or an alpha helix as the singular form, and beta pleated sheets. These are held together by hydrogen bonds. So you can see that they are still chains of amino acids, but different hydrogen bonds are also formed, creating their structure of the sheet or the helix. The tertiary structure is the unique 3D structure of the protein. This is due to chemical interactions among R groups of the amino acids, either repelling each other or attracting each other to form these unique twists and turns of the protein. The quaternary structure is formed by weak interactions between polypeptide subunits. This consists of more than one amino acid chain, so there should be more than one primary structure to form a quaternary structure. So here's a recap of all of the protein structures from primary to quaternary. We talked a little bit about denaturation when we first started proteins. In denaturation, the protein loses its shape without losing its primary structure. This can be reversible or irreversible. Sometimes by just taking the protein out of this non-ideal condition, it can reverse that denaturation and turn that protein back on. But sometimes it's permanently changed. And a good example of that is the albumin protein found in egg whites. Once you start to cook the egg, you can see that the protein changes color from clear to white. If you were to leave this egg in the pan, the whiteness of the egg would not return to this clear color. It's permanently changed. So that change, that denaturation of albumin, is irreversible. When the folding of protein occurs, to help keep it in its ideal shape, chaperonins are needed, also known as chaperone proteins. They're absolutely necessary for protein function. Lastly, of our macromolecules, we have nucleic acids. Our two major types are deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. These are also known as polynucleotides because they're made up of the monomer nucleotides. They carry the genetic code, specifically DNA. RNA's job is going to be to make proteins, and DNA's job is going to be to carry that genetic code. Nucleotides are made up of nitrogenous bases, pentose sugars, and phosphate groups. Nitrogenous bases can be broken down into pyrimidates and purines. The way that I was taught to remember this in school was all girls are pure. To remember that adenine and guanine are purines. Cytosine, thymine, and uracil are all pyrimidines, and the specific bonds that link nucleotides together to form our polynucleotides or nucleic acids are phosphodiester linkages. DNA is going to carry the genetic information for single-celled bacteria and also multicellular animals. The entire genetic code that is found within DNA is called the genome. The study of that is genomics. Eukaryotes, or multicellular organisms, have chromatin, which is the building block for chromosomes, which hold those genes. Chromosomes can carry tens of thousands of genes. And DNA can turn genes on or off, depending upon different environmental factors. DNA has this double helix structure. I'm sure you've seen this before. Oftentimes this is compared to a ladder kind of being twisted. So the handles of the ladder are made up of a sugar and phosphate backbone, and the ladder rungs are made up of these nitrogenous bases. So there's going to be a nitrogenous base on either side, and it's going to want to pair with a specific nitrogenous base on the other side. So remember, we listed 5 nitrogenous bases that can be found within nucleic acids. Only 4 of those are found in DNA: cytosine, guanine, adenine, and thymine. Adenine and thymine only pair with each other in DNA. Cytosine and guanine only pair with each other in DNA. So C and G pair, A and T pair. So when DNA is replicating, it's going to form a complementary strand that will have its complementary base pair on the other side. A will find T, G will find C. RNA is involved in protein synthesis. DNA kind of provides the instructions for that. There are 4 different types of RNA. There's messenger RNA or mRNA, and that's going to facilitate transcription. So it will take that complementary strand of DNA and form a strand of RNA that matches it. When it does so, RNA only has 4 different types of nitrogenous bases as well. It has adenine, guanine, cytosine, and uracil, no thymine. So in RNA cases, adenine likes to pair with uracil. G and C still pair. So when messenger RNA transcribes that DNA into RNA, it does so that it can leave the nucleus. DNA is much too large to leave the nucleus, but RNA can. And we'll talk about that more also next week when we get into the cell. Transfer RNA or tRNA is going to facilitate translation. So translating that RNA sequence from mRNA into proteins. Ribosomal RNA or rRNA is where the mRNA will bind on the ribosome. The ribosome is the place where proteins are created. And microRNA or miRNA, one that you don't hear about too often, has a role in gene expression. So it can interfere with mRNA messages. And here's a table that compares DNA versus RNA. There are a lot of differences. So let me know if you have any questions regarding that lecture for the week.